Yes, both of you switch on your videos. Yeah. Now tell me, in the previous class, we were discussing about the microsporogenesis, right? The formation of the microspore, isn't it? Yes or no? Yes, ma'am, we were. No, ma'am, we weren't. No, ma'am, we don't remember.
Ma'am, what
Say
did you ask?
I am asking, in the previous class, we had left off till microsporogenesis, isn't it? So, moving forward, now see, microsporogenesis results into the formation of the microspore. Microspore meaning the pollen grain, right? And pollen grain is the male gametophyte or the male gamete of the plants. Now, further, when we talk about the pollen grains, so they are mostly spherical in shape, but it's not necessary that they are, like spherical in shape for every species. Microspores or pollen grain, they are spherical, they can be, you can say ovoid, they can be oval, round shape, they can be star shaped, different kinds of pollen grains are there. Now, the diameter of the pollen grain is usually 25 to 50 micrometer, and every pollen grain is surrounded by two layers. I think this was also done, isn't it? Tube cell, generative cell, I believe this was also done. Pollen viability, I've also discussed, like how they are valid for a certain period of time, like 30 minutes for this particular rice and wheat. Have I discussed this?
No, ma'am.
No. And what about pollen allergy?
Yes, ma'am.
Okay. So talking about pollen viability. Now see, once the pollen grains are made inside the anther and now they are released. So once the pollen grain are released, they are effective only for a period of time. That period of time for which the pollen grains are effective, we call it as the pollen viability or the validity of the pollen. So pollens, once they have been shed, they have been released, they have to land on the stigma, on the right type of stigma before they lose their effectiveness. And the time period for which they are viable, for which they are, you can say, effective, is called as the pollen viability. Now, apart from the species being, like, this particular time period being specific for species, it also depends upon the temperature and it also depends upon the humidity. So when we talk about the pollen viability in some cereal plants, such as the rice and the wheat, it is 30 minutes after they are shed. Meaning within 30 minutes, they have to land on the stigma or else they'll be useless. Whereas for some other pollen grains, like from the members of family Rosaceae, Leguminosae, Solanaceae, these different families of plants, so their pollen grains, once they are shed, they are viable for several months. So it like the time period is not 30 minutes that they'd have to land on the stigma within 30 minutes for the proper, you can say, fertilization to take place. No. This is clear, pollen viability?
Yes.
Okay, very good. After that, there comes cryopreservation. Now, you people might have heard about students. I believe I've taught you this also. No, no, no. So talking about cryopreservation, so it is a method wherein you can store pollen grains for future use, or you can store them for the use when that particular plant is not of that season. Right, now how do you store your pollen grains? You store them in liquid nitrogen at 196 minus 196 degrees Celsius. This process of preserving pollen grains is called as cryopreservation. You're basically storing it at a very, very low temperature in liquid nitrogen. Now, just like how seeds are available throughout the year, same way, when you talk about the pollen grains, they are also available because you're preserving them, you're storing them. This storage, like this particular storage of the pollen grain, It is used as pollen bank, wherein you have different kinds of pollen grains present. And it is very similar to the seed banks. Now, the cryopreservation, it is used in crop breeding programs, right? So whenever there will be the need of any particular type of pollen grain, the people can retrieve it. Why? Because it has been preserved. So this was about the male part of the flower, everything associated with it. Now we're going to talk about the female gametophyte, the female reproductive part of the plant, that is the gynoecium. Now one unit of gynoecium is called as the pistil or the cup. Now if the gynoecium, if the female reproductive, there's only one pistil present in a flower or in a plant, we call it as monocarpellary. Mono means one, carpellary means referring to the carpel. So when there is only one carpel, we call it as monocarpellary. And when there is more than one pistil or more than one carpel present, we call it as multicarpellary. Now, I believe all of you know already that what like the two basic types of cell in any organism's body are somatic cells, which are called as vegetative cells in the case of plant. They are usually diploid, and the germ cells, that are the reproductive cells, which are haploid. So
Ma'am,
remember, hmm,
I think we finished this. We took this in the last class.
yeah, because I believe that too. Because I was like, I'm repeating these things. I believe this is also done.
Yes, ma'am.
The
Pre-fertilization, I think.
I have to start from here. Megasporangia was not done, right? I don't remember teaching you this thing, isn't it? These things I have not taught. So I'll start from here. The megasporangium. Now, megasporangium is a very fancy name for ovule. Now, when we talk about the ovule, it is a small structure which is attached to the placenta part of the ovary, and it is basically attached to it with the help of a stalk-like structure which is called as a funicle. You can see the funicle in this particular picture. There is the funicle, this area. Now, this is the part which is attached to the placenta. Now, the junction between the ovule and the funicle, there's a line which is called as the hilum. Okay, hilum. Now, remember, whenever you see the picture of an ovule, if if it is like this or if it is inverted, always remember the part that will be called as the basal part, right? The base part is going to be chalaza. No matter whether it is up, whether it is down, it doesn't matter. Keep it in your mind that chalazal end is your basal end and micropyle end is the top. This must be fit in your mind like a puzzle. Like puzzle piece that is fixed forever and glued into your mind. Always remember chalazal side is the basal side, micropyle side is the tip side, okay, the top side of the apical portion. Now, in the ovule, there is the presence of protective layers that are called as integument. So, there is an outer integument and there is an inner integument. Two layers are there which protects the particular embryo sac. Okay. Now, the integument, like the part of the integument which is not present at one area, we call that area or we call that region as the micropyle region. Always remember, micropyle region is the top portion. It is the small opening from where the pollen tube enters the ovule. This is the point from where the pollen tube will enter. Opposite, right opposite to the micropylar end is the chalazal end, which is the basal part of the ovule. Now, inside the megasporangium, there is the presence of nucellus. This particular tissue, the light blue one, this whole entire area, this is the nucellus and it is a nutritive tissue around the embryo sac. Okay, it's responsible for the nutritive purposes. Okay, so yeah, now the cells of the nucellus, how are they nutritive? Because they have abundant food reserve in them. Because of that, they are a storehouse of nutrients. Now, inside the nucellus, there is the presence of the embryo sac, which is the female gametophyte. And one ovule generally has one embryo sac present. Okay, is it clear?
Yes.
No. Now, basically, talking about the megasporogenesis. Now, megasporogenesis, it is basically the process of formation of megaspore from megaspore mother cell. Okay, just like how microspore mother cells gave rise to the microspores, same way here, megaspore mother cell will give rise to the megaspore. And the process will be called as megasporogenesis. Megasporogenesis. So, basically, this process or the megaspore mother cell, it gets differentiated in the micropylar area, wherein these megaspore mother cells, they have a lot of, you can say, cytoplasm, and they have a prominent nucleus, and they start to undergo the process of meiosis in order to form four haploid megaspores. Now, out of these four haploid megaspores, three of them degenerate and only one is functional. This functional megaspore will give rise to the embryo sac. Do you see this egg-like structure right in the middle? This one? This is the embryo sac, which is formed from that one functional megaspore. Now, since this whole entire development is happening because of a single megaspore, we call it as monosporic development. Okay. Now, let us see how the formation of embryo sac takes place. So, the nucleus of the functional megaspore, that one functional megaspore, first of all, it starts to divide by mitosis. Mitosis basically means to increase the number, right? So, it forms two nuclei, and those two nuclei, they start going towards the opposite poles. Okay. Now, further two more mitotic divisions take place. So, from two, it becomes four nucleated. And then furthermore, then eight nucleated. So, all in all, eight nuclei are made inside one cell. One cell is there, eight nuclei are. This type of mitotic division is called as free nuclear division, wherein nuclear division is happening, but it is not being followed by cellular division. So, for example, always remember one thing, like for example, if this is a cell, inside there is a nucleus, okay? So, when the nucleus would divide, like during cell division, first the nucleus divides and forms two nuclei like this, one and two, okay? Then every nuclear division is followed by the division of the cell called as the cellular division or cytokinesis. Okay, so this will result into the formation of two cells. Okay, but here in the megaspore development, we are seeing this is one cell, and inside the one cell, first two daughter cells are formed, then four daughter cells are formed, and then totally eight daughter cells are formed. Okay, so now these eight daughter cells, like nuclei, are there inside one cell. So this is a free nuclear division, meaning every nuclear division is not being followed up by the cellular division. So this is what we call as free nuclear division. Now after that, cell walls start to form inside that one cell. Now you will understand it better once you see in this particular diagram. See, wait, let me change the color of the image. All right. So there is a megaspore mother cell, which is diploid cell. This circular structure that you see, this one is a megaspore mother cell. Now understand this very carefully, okay? Now this one megaspore is undergoing meiotic division, and you people know that at the end of meiosis, four haploid daughter cells are made. So here also four haploid means one set of chromosomes are there. So four haploid megaspores are formed. Clear? This much is clear? Keep on saying yes so that I'll
Yes.
know. Then out of these four megaspore, three of them are useless. They are going to degenerate. These are degenerated, and there is only one functional megaspore. Okay. Now this one function, now you don't have to reduce the number of chromosomes. You already have a haploid set. Now they will start to increase the number of nuclei. So now it will undergo the first mitotic division and form 1 and 2 nuclei. Then these 2 nuclei are undergoing the second mitotic division and they are going to form 1, 2, 3 and 4. 4 nuclei. Then these 4 are undergoing and they'll ultimately form the 8 nuclei in one cell. Now, those nuclei will start to get arranged in the cell after they have cell wall formed. Now, do you see these yellow, like red in the middle and yellow on the outside? Do you see that? So, those yellow parts, they denote cell. The red part denotes nucleus. Okay. So, they have arranged themselves in such a way. Okay. Now, we will study their arrangement. Now see, out of the 8 nuclei that were formed, 6 are surrounded by the cell walls and made into the cells. The remaining 2 become the polar nuclei. Now this is a clearer picture of the embryo sac wherein you would understand it better. See here, basically what happens is that, see, do you see the, like, they say there is a statement that says that a mature embryo sac is 7 cell, 8 nucleated. Okay. They say that there are 8 nuclei present, but only 7 cells. How? Now see, all these lines that are there, it denotes a cell wall. Okay. A cell wall denotes that a cell is made. Okay. So we have cell number 1, cell number 2, cell number 3 on top. I mean, not on top, it is a chelazal line. Then we have cell number 4, cell number 5 and cell number 6. And do you see this one, the bigger one? This is the 7th cell. So we have 7 cells. Now count the red ones. So there's 1 red, 2 red, 3, 4 and 5, 6 and 7 and 8. So you have 8 nuclei arranged into 7 cells. So you'd have to draw this thing to justify your answer that how is a mature embryo sac 7 cells and 8 nucleated. It's very weird to think about it, right? This is how it's arranged. Now I'll tell you how each of the nuclei that we saw, how they are getting arranged at different areas. Now see, distribution of the cells in the mature embryo sac. 3 cells are present at the micropyle end, meaning this end. Okay, this part. So 3 cells are present at that part. Okay, they form the egg apparatus. So these 3 cells together, they are making the egg apparatus, in which the 2 cells at the below, the 2 large cells, they are called as the synergids. And then the one that is behind it is the egg cell. So 2 synergids, 1 egg cell together, they make the egg apparatus. Now, do you see this lettuce leaf looking structure? This one? This is called as the finger-like structure called as the filiform apparatus. Why is it there? It's there to guide the pollen tube. Okay, that's why it's there. Now, talking about it. So 3 cells get arranged at the micropyle end and they are called as the egg apparatus. The rest of the 3 cells, they arrange themselves at the basal end, that is chelazal end, and they get called as the antipodal cell. Now, 336, now we are left with 2 nuclei. Now, 2 nuclei are present right in the middle of the cell and they constitute a part of the, you can say, central cell. Because that one cell in the middle, the 7th one that I drew, that is the central cell which has 2 polar nuclei in it. Is it clear, students? The basic arrangement is clear?
Yes.
So always remember that a typical angiosperm embryo sac, like at maturity, it has 8 nuclei, but it has 7 cells associated with it. This is very important and you must know how to justify this particular state. Is it clear to everyone?
Yes.
Any doubts? Anything you want to ask?
No.
Now the next thing that we have, now all the pre-fertilization events that were taking place, those are done. Now the male gamete is made, the female gamete is made. Now what we need to do? We need to make the male and the female gamete meet each other for the purpose of fertilization. So the next topic that we are going to see is pollination. So basically, when we talk about the pollination, it is the transfer of the pollen grains from the anther to the stigma. Okay, so from the anther of the male gametophyte to the female part, that is the stigma. That is what we call as pollination. So it is the transfer of pollen grains from the anther to the stigma. Okay, the process is called as pollination. Now, the different types of pollinations that are there, it depends upon the source of pollen. Source of pollen means from where is the pollen grain coming. So there are three types of pollination. Autogamy, geitonogamy and xenogamy. Three types of pollinations are there. So when we talk about auto means self. So autogamy is also called as self-pollination. So this is the pollination wherein the pollen grains are transferred from the anther to the stigma of the same flower. So for those flowers that have the stigma and the meaning the pistil and the stamen in one, for them self-pollination takes place. Meaning in the case of bisexual flowers, self-pollination happens. So some plants such as viola, which is known as common pansy, there's oxalis and commelina. These three plants, they produce two types of flowers. One is a flower that is open and exposed, meaning all you can see the stamen, you can see the carpel. Another kind of flower are the ones that are closed. You can't see the stamen, you can't see the carpel. So, when we talk about the cleistogamous flower, they are closed. So, anther and stigma, they are very close to each other. So, whenever the pollen grains will be released, it will land on the stigma and successful pollination will be done. So whenever the anther will dehisce, meaning the anther will burst, the pollen grains will come directly in contact with the stigma. And in the case of, you can say self-pollination, you can say in the flowers that are closed, there can be no other kind of pollination taking place other than the self-pollination. But what is the advantage? The advantage is that there are no pollen grains that are wasted. Now imagine the anther is there, the anther is going to burst. When the anther is bursting, it's going to release the pollen grains everywhere. So during that, some pollen grains can be wasted also. But in the case of a cleistogamous flower, the flower that is closed, if the anther is bursting here inside, no wastage. So in them, assured seed set is produced even in the absence of pollinator. They don't need any agent to cause pollination. The anthers dehisce, pollination takes place. So autogamy is clear to everyone.
Yes.
What about the others? And why are your
Yes.
videos on? Switch on your video, students. Muniba, switch on your video. Okay, so after autogamy, there comes the next type of pollination called as geitonogamy, which is sort of like cross-pollination, but not actually. See, in this case, the transfer of pollen grains takes place from one flower to another flower, but the plant is same. So if I talk about the genetically nature or genetic nature of the pollen, will it be same or different? It's coming from the same plant. So will it be same or different?
Same.
Same, right? Because the plant is same even though the flowers are different. So when we talk about the geitonogamy, so in this case, the transfer of the pollen grain takes place from the anther to the stigma of another flower. Like from one flower to another flower, but the same plant. So functionally, it looks like this is cross-pollination, but genetically it is self-pollination only. Now, furthermore, the third one, that is actual cross-pollination, called as xenogamy. This is the case wherein the transfer of the pollen grain is taking place from one plant to another. Whereas geitonogamy was this, one flower to another in the same plant. Okay. So now when we talk about the cross-pollination, so here genetically different type of pollen grains are brought to the stigma, right? So these are the three types of pollinations that you will have to study. After that, there comes one more thing. Now, when we are talking about cross-pollination, now obviously the pollen grains cannot fly. So they need something to carry them to the stigma. So we come to the concept of pollinators. The agents that cause pollination, we call them as pollinators. They are of two types, living and non-living. Living pollinators are called as biotic pollinators. Non-living pollinators are called as abiotic pollinators. So non-living pollinators are wind and water, whereas the living pollinators are different organisms, insects, animals. But remember, majority of the pollination that takes place, it takes place because of the biotic pollinators and few because of the abiotic pollinators. So first we are going to talk about the abiotic one. So in this case, since the wind and the water, they do not move according to any organism, right? They just flow the way they want. So here, a successful pollination taking place is a chance process. It's a luck factor, right? If the wind blew in that direction where the pollen had to be dispersed, then that is a chance factor and luck is in the favor. So for this, for the compensation of this particular chance factor, what these plants do, like those plants that get pollinated by the wind or the water, they produce a large number of pollens just to compensate for the waste of pollens that might take place. And amongst the abiotic type of pollination, the pollination by the wind is very common. Water pollination is rare, and the pollination with the help of wind is called as anemophily. So for wind pollination, the pollen grains, they have to be very lightweight so that they can easily be carried away with the wind. They have to be non-sticky. And in that case, the stamens have to be exposed so that the pollen grains directly come in contact with the air or the wind. Apart from that, the female reproductive part should be feathery so that they can catch the airborne pollen. Now, usually the flowers that are pollinated by the wind, they have a single ovule, usually, and several flowers are packed into inflorescence in them. One such example is the corn cob. How many of you have seen corn with the whole entire covering? The layers, the green covering on it, right? So if you remove that covering, do you see hair-like structures, yellow, yellow? Those are the tassels that are the stigma and the style of the plant. Okay. So remember, pollination by the wind, which is anemophily, is very common in the case of grasses. The second is the case of pollination by water, that is hydrophily. So when we talk about the pollination by water, so this is rare and usually 30 genera, they have this type of pollination in them. So basically for the transportation here, the male gametes, like basically it's for the aquatic plants like bryophytes, pteridophytes, algae, where water is required for fertilization. So some of the water pollinated plants such as Vallisneria and Hydrilla are freshwater plants. Then there is Zostera, that is a marine plant. So let us see, we have unique pollination types. So in the case of Vallisneria, the female flower, they reach the surface of the water, okay, and the male, like you can say the pollen grains, they are released on the surface of water. So like this is water surface, the pollen grains will be released over here. The female flower will just come on the surface of the water and get fertilized that way. So this happens in the case of Vallisneria. Okay, because they have the female flower is long. They have a long stalk. Second is the case of Zostera, which is a seagrass which is found in the marine water. So here, female flowers are submerged. This is the level of water. Female flowers are here, submerged in the water, and pollen grains are released inside the water. Now here the pollen grains are long and they are ribbon-like. Okay. And now since here, in either of the two cases, the pollen grains are being shed into the water. So they have to have something to protect them from getting wet. So they have a mucilage covering present on top of them, which sort of acts like a raincoat. Imagine like that. It protects them from getting wet. Now always remember that the majority of the aquatic plants are not pollinated by water only. It doesn't mean that if they live in water, they'll be pollinated by water. No. Majority of the aquatic plants, such as water hyacinth, water lily, these plants, they emerge at the level of water and then they are pollinated by the wind or by the insect. Now, one such question comes to mind that the wind and the water pollinated flowers, they are not brightly colored, they're not beautiful. Can someone tell me why? Why so?
Because it doesn't need to be pollinated by insects.
Speak a little louder, Safina. Speak loudly.
Mom, it doesn't need to be pollinated by insects, where insects are attracted to colored flowers.
Since wind and water, they do not need insects, right? So they don't even have to attract insects. So since they don't even have to attract insects, so they don't even do the extra work. They're like, okay, we'll just look like this. We'll not dress up for the insect. And thereby, because wind and water do not see the dressing, that's fine. Understood? Now, majority of the flowering plants, they are pollinated by biotic agents. Biotic means living agents such as bees, butterflies, different birds, bats, flies, beetles, moths, ants. These are some of the, you can say, biotic agents, amongst which honeybees are the most dominating and the most common type of pollinating agent. Now see, pollination by wind was anemophily, pollination by water was hydrophily, pollination by insect is called as entomophily. Okay. Now, in this case, larger animals also act like a pollinating agent, such as primates like lemurs, then arboreal rodent. Arboreal means they live in the trees. They can also cause pollination, and reptiles such as the lizards, they can also cause pollination. Now, those insect pollinated flowers, they'll be large, they'll be beautiful, they'll be colorful, fragrant, and rich in nectar. These are the adaptations of the flower so that they can attract the animals, attract the insects. So, majorly, when the flowers are small in any plant, then they'll be packed into a bunch, which we call as inflorescence. And then they will attract the insects by the fragrance or by the color. But one such thing you should remember, for those flies and beetles as pollinators, now those plants, they do not emit out fragrance. Rather, they emit out foul odor, meaning bad smell, so that they want to attract the flies and the beetles. Understood? And now all of these things, they offer it as a reward to the pollinators, like nectar, pollen grains. So like honeybees, they go to the flower for nectar so that they can make honey and thereby cause pollination also, because they move from one flower to another, thereby causing pollination. So whenever the insects, they come to the flower to take the nectar and other rewards, so the bodies of such animals, they get coated with pollen grains. And when these animals, they come in contact with the stigma of another flower, the pollination successfully takes place. Is it clear, students?
Yes.
Okay. Now, in some species, the plants, they offer floral rewards in a different manner. meaning they will provide such as a safe place to lay the eggs, such as Amorphophallus, which is a very tall flower. It is 6 feet in height, and it provides a safe place for the insect to lay their eggs. There's Pronuba moth and Yucca plant. They can't complete their life cycle without each other. They are very dependent on each other. Like Pronuba moth requires yucca plant, which is a very tall plant, to deposit its egg. And yucca plant needs the moth to pollinate it. They can't live without each other. And once the moth has deposited its egg in the yucca plant, then the eggs, when they hatch and the larva comes out, so it comes out and it doesn't harm the plant. Okay, so as soon as the seed starts developing, the larva will move out. Now, there are some organisms that take the pollen reward and do not help in pollination. We call them as pollen robbers. They do robbery. Okay, because they're not doing anything for the plant, but they're stealing everything. We call them as pollen robbers. Any doubts up until now, students? Okay. Now tell me, what is the last thing you people wrote down? You write a little bit, then we'll move on to the next topic. What is the last thing you people wrote?
Pollen grain, male gametophyte.
Male gametophyte.
Pollen grain, ma'am.
Can't hear. Speak loudly.
Yes, ma'am, there.
This part. Okay. All right. No doubt.
Done, ma'am. Done. Done, ma'am.
1 second.
Okay, sure. No problem.
Done, ma'am.
Done.
Done, ma'am.
Both of you are done, students?
Yes, ma'am.
Done, ma'am.
Mhm.
Done, ma'am.
Done, students? Done till here?
I'm writing gynaecium part.
Oh.
Done.
Done, student?
No, ma'am. Done, ma'am. Done, ma'am.
All right, students.